Hello, everyone. We're still alive. Keep some energy going. I know it's a long day. I know many of you have given presentations and are a little winded, but we can make it, and there's still more to come. And so I'd like to welcome everyone for the keynote address today. We have our own acting VPR, Dr. Ilhem Messaoudi. I believe she's going to talk about her research journey. Her journey to research leadership, sorry. So I'm not going to go through a long introduction, but I do want to say that this symposium is in part sponsored by the Office of the Vice President for Research, and so we really appreciate the support. And so thank you, and I'm going to give you the stage. They're not here to listen to me, they're here to listen to you. So.
Thank you. Well, good afternoon, everyone. I do understand that I stand between you and reception. I heard the word reception. Are there libations? And there's libations at the reception. So I will be very conscious of the time. I'm actually going to do a stopwatch so I can make sure that I tend to be long-winded sometimes. So it is absolutely an honor to be standing in front of you today. Thank you, Joseph, for inviting me to share a little bit about my research and my journey. I take no credit for this title. My postdoc, Heather, decided that this would be a really good summary of my journey. I do love viruses. I have no vices, of course. I'm perfect. And maybe some leadership wisdom to impart today. So I'm going to try to sprinkle some of this in between some anecdotes of research. And I'm hoping to leave some time for some discussion at the end. So first, I want to tell you a little bit about who I am. So I was born in Tunisia. It's a little country in North Africa. I'm going to try to do this, maybe. Which one is the laser pointer? There's only 2 buttons on this thing. That goes back, that goes forward. Nope. All right, well, you're just gonna have to trust me while I figure this out. So I was born in Tunisia, it's in North Africa. And I was born in Carthage, and that's a picture of Carthage. It's a very beautiful market. And so our claim to fame, amongst many things, olive oil, some interesting wines, beaches, tourism, the only second Colosseum in the entire world after the one in Rome, is Tatooine. So for all of you who are old enough to have watched Star Wars, okay, great, thank you. There's the picture of Star Wars, the Tatooine in Star Wars, and then the real Tatooine above it, which is very close to where my dad was born, near the Atlas Mountains. And then when I was a teenager, my family moved to Oman in the Middle East. And we lived in Muscat for a few years. That is in the Sultanate of Oman. And then we moved to Abu Dhabi, which is the United Arab Emirates right next to Oman. I first started as a homeschooled at the French school, which basically meant all 5 kids who were trying to have an education that was in line with what was happening in France got together. There were multiple people who came in and out of the house where we all hung out. And a lot of long-distance learning, a lot of sending exams to the embassy to get graded. And then my last 2 years, I took English at night school at the British Council and got my O-levels in English and transferred to the American British Academy and got an international baccalaureate degree, which back in the day, I'm not going to tell you what year this is because I'm not dating myself. This was a big deal. Now the International Baccalaureate is a pretty well-established program. But the reason I did that is because I've always wanted to be a microbiologist. I know it sounds really geeky, really nerdy, very cheesy, but it's the truth. I've wanted to be Louis Pasteur since I was a small kid. I just thought the whole idea of microbiology and these little invisible things that made you sick and vaccines and therapeutics was just fascinating. And so I wanted to pursue this dream, and I decided that the best place to pursue the dream was to come to America, which meant that you had to learn English. You had to get a degree that you can actually convert. And so that's why I did what I did. And I landed in Lafayette College, which is in Easton, Pennsylvania. It's a small town on the Jersey, Pennsylvania. And it was great. It's right on the Lehigh River, where the Lehigh River meets the Delaware River. I only know this because in my infinite wisdom, when it was nice and warm in the fall, I decided to join the rowing team. Nobody told me that at some point I was going to have to row where there's like little ice chunks floating by. But it was a wonderful experience. And I got my first bite of what research really was in the lab of Dr. David Husick, who's now a professor emeriti, and I can't even think about that because he was still a pre-tenure when I joined his lab. And I got to work on this really cool project looking at carbonic anhydrase enzyme and trying to isolate the one that was not living in the cytoplasm, but the one that was actually integrated into the mitochondria of algae. It was great. I got to grow large liters of algae for many, many months, and tried and failed for about 2 years to extract this enzyme. But I learned a lot of things about research, which is, gotta love what you do. You're more dogged than the experiment. And thinking about alternative ways and troubleshooting and how to present a poster and how to think through your project. And it was a phenomenal experience that set me up to go to Cornell Medical School to get a PhD in biomedical sciences. So I think being a graduate student, 20 years old in Manhattan, was probably the next best thing that happened to me after being a leopard for 3 years and on the rowing team and getting to do all that. And this is where I really started to pursue my true love. I mean, algae were cool, don't get me wrong. They were cool, but 2 years of growing liters of algae and failing at isolating one enzyme was good enough, and I was ready to move on to something else. So I joined the laboratory of Dr. Janko Nikolic-Zugic at Memorial Sloan Kettering. One of the cool things about this program is multidisciplinary. It spans multiple institutions. So you could rotate actually in any lab that you wanted, from Rockefeller University, Hospital of Special Surgery, Memorial Sloan Kettering, Cornell itself. I think I remember it was something like 600 laboratories that you got to choose from. So the world was truly your oyster. It was just wonderful experience. I worked in a lab that was really interested in understanding how T cells got activated. So T cells are really important lymphocytes in our body. They patrol. I always tell people that the T cells are the Navy SEALs of the immune system. You don't call upon them unless you have to, because once you do, there's no turning back until the enemy has been eliminated. And I studied how small changes in the molecules that present antigens to T cells could have dramatic impact on the outcomes of disease. This was also, again, just being at Memorial Sloan Kettering, surrounded by brilliant people. I think one of the most impressive experiences that I had was showing up for my first biochem class as a first-year graduate student, and Professor Darnell was lecturing, and I know maybe if some of you are old enough to remember Darnell's biochem book, but I remember just looking at the book and being like, Darnell, is that the same guy? And the answer was yes. So I had 7 Nobel laureates lecture us in class, and they just hung out at the bar on Friday and would sit down next to graduate students and talk about science for hours. So amazing experience, really just for me resonated with everything I thought graduate school would be about. I worked really hard on understanding CD8 T cells, showed how the changes in the floor of major histocompatibility class 1 molecules, which present peptides to T cells. So one cool fact, your T cells can tell the difference between your self-antigens and a foreign antigen based on 3 amino acids. That's it, 3 amino acid residues will tell them the difference between a flu peptide and your myelin, for example. And so when things go wrong, they go really wrong, and that's why I call them the Navy SEALs of the immune system. I was really fortunate. My work at the end culminated in a science paper, so I was very, very happy camper. And I decided to follow my PI to the other side of the country. So we landed in Portland. And again, maybe this crowd is too young, but any of you watch Portlandia? Yes, Portland is Portlandia. That is not an exaggeration. So people walk around with stickers on their car that says, "Keep Portland weird." It's a thing. You eat donuts that are really strange. It's a thing. You go hiking, lots of Subarus, total granola country, amazing experience. So I'm going to pause here because this was my postdoc time. And there is nothing that will ever come close to being a postdoc. I know that you may not be feeling that way right now, but being a postdoc was the best time of my life as a researcher. I had zero obligations, right? Didn't have to write a grant, didn't have to write a protocol, didn't have to mentor a graduate student unless I wanted to. Just got to focus on my science. There was no qualifying exam, no mentoring committee, no every 6 months somebody's meeting with you to ask you about your progress. I mean, somebody is. But you get to set your pace. You get to have the time of your life. So enjoy it. Postdoc years are the best time. Just enjoy the fact that you get to set the pace. You get to think about your project. You've gotten a lot of responsibility, a lot of freedom. Nobody's micromanaging you. And think big and think wide and figure out what it is that you really want to do when you grow up. So for me, I did not know what I really wanted to focus on. I still want to be Louis Pasteur. So how do I get there? Study T cells, like check. All right, maybe you should study some microbes now so I could get that host microbe interaction going. But the thing that really struck me as a postdoc is I followed my PI from Memorial to Oregon Health and Science University, but his appointment was actually at the Oregon National Primate Research Center. And never in my whole career did I think about doing work in non-human primates. I did not even think about that as an animal model, did not know anything about it. And it was the heydays of really trying to understand SIV, so HIV research was booming. How do we use this animal model where there is a virus that looks like the human virus, causes the same disease in monkeys, and how do we use this model to really ask some difficult questions that we could not do in the human system? And it opened my mind to the fact that there are different times where you're going to call upon different models to ask your scientific and answer your scientific questions. Sometimes it's a cell line. Sometimes it's going to be a three-dimensional organoid system. Sometimes it's going to be a mouse, and sometimes it might have to be a non-human primate. And every model's got its place in the world of research, and you got to make those decisions really carefully and really methodically. You don't jump into a non-human primate model without having a really good reason to do those studies. So I learned how to handle non-human primates. So anybody complaining to me about doing a mouse injection, come see me afterwards. I got a certificate in animal handling, and I learned how to do non-human primate handling actually at a very high containment level. It was great. I got to pursue my love of T cells, superimpose on that my love of what happens when the immune system gets old, which I developed at the end of my PhD. So I started with trying to understand antiviral T cell responses, and then became really interested in why do older people not mount really good antiviral immune responses. Was able to then pursue that question in a non-human primate model, which had a lot of great similarities to human physiology. And used this model to look at things like caloric restriction and how caloric restriction can delay aging. Is there an optimal window to start caloric restriction? What are the impacts on the innate versus the adaptive immune system? Got to learn a ton of things. Was one of the people to contribute to a resource library for studying things in non-human primates, because at the time that we did this, we actually did not have a good, you couldn't go to like any antibody company and pick out an antibody that you knew would work in a non-human primate. We had to validate a lot of that work. So learned how to work in large teams, how to work in a space where there was a lot of unknowns, and be comfortable with being sort of on that leading edge of questions. And towards the end of my postdoc, I became kind of full circle back to that love of microbes. And I started getting really into emerging infectious diseases. And so I got a job at Tulane Primate Center. And my job was supposed to start September 2nd, 2005. And my job disappeared because Hurricane Katrina hit New Orleans 3 days before I was supposed to start my job. I'd moved. My husband and I were newlyweds. We bought a house. This is actually a picture of our house. 134 Murat Street, I still remember it. It's an old duplex that was built in 1904. So the good news was that because it was built in 1904, it has survived 15 other hurricanes and was still standing after Hurricane Katrina came through. Bad news is nothing else survived in New Orleans. But it taught me a lot of resiliency. That was an experience that completely changes your life, right? You imagine you have a dream, you're getting your first faculty position, you're ready to go, you have a plan for what you wanted to study. I had a mentor lined up because I was recruited as part of their COBRI, which is a grant that we have here. It's grants that a lot of idea states can apply for. They have faculty slots on them, and they're targeted at states that are below the NIH funding percentage that you can see in the other states, like the coasts. So Louisiana, as well as Kentucky and Mississippi, Alabama, et cetera, can qualify for that. So a lot of things to learn from having your life completely turned upside down and having to take a year off from research. And the reason I share this is, A, we just had the 20th anniversary of Katrina, which always reminds me that I've been married for 21 years, and I can't believe that. But also just that you can come back from things like this. You can have a hurricane come through and completely destroy everything that you thought that you were going to do. And you can bounce back. You can bounce back with support from family, from friends, from people you have built, from that research community that you have built over the years that you were a graduate student and postdoc. So don't ever forget that the people you come across in all the stages of your life will have a role to play in your life at some point. So nurture those relationships, spend time, invest in the people you meet, because you never know when you'll need to call upon them to help you. So I happened to come back. So what do? Your house is, well, still standing, but there's really interesting orange mold growing from the ceiling. My husband had a spot in Tulane, but they moved over to Houston. So we had to deal with having him transfer medical schools. And so I went back to OHSU because I had such strong ties to that community that they opened their arms, and Scott Wong, whose picture is not here, opened his arms and took me in as first as a postdoc, a second postdoc, and then promoted me to a research assistant professor. So non-tenure, complete soft money, but allowed me in those first 6 months I was postdoc to write for grants on my own. And so I did. It was definitely, I had to hustle. I went in as co-I on other grants to provide expertise in immunology and worked a lot of long hours to write my own proposals in. But I had a couple of grants when I decided to go back on the job market. My husband was finishing his second year of medical school, so that's a good time to transfer. We went back on the job market, and I was really lucky. Nancy Hagewood, whose picture is featured here, had just been hired as the Primate Center director. And she said, well, why would you leave? And I said, because I'm on a real tenure track position. This whole research assistant, year-to-year contract, 100% soft money is not really what I envisioned I could do with myself. And so she was like, well, we're looking for immunologists to stay. And by then, my prior postdoc mentor had left OHSU. She's like, we have a void, you can fill it. So my other thing is never, ever walk away without thinking about the possibility of an opportunity opening up. Again, that relationship building, making sure your family is bigger than the lab you're in. Your family has to be everyone in that institution that's around you. Because if I had only been connected to the people in my lab, I would not have gotten this faculty position. So I got a real faculty tenure track position in 2008. And then I spent a few years at OHSU, then went down to University of California system where I spent 10 years, both at UC Riverside and UC Irvine, and then came here almost 4 years ago. So I'm gonna tell you a little bit of what we did along the way. Over that period of time, so I guess you can kind of do the math yourselves. Over that period of time, I have grown this research program that's really centered around immune dysregulation. So what dysregulates our immune system? And the ultimate goal is understanding how the immune system is dysregulated can give us keys to what can we do to stop that dysregulation or intervene. What are the pathways that we need to activate or deactivate? What are small molecules that we can test? What are the big questions that we need to be pursuing? And so my lab today focuses on aging and immunity. It focuses on still viral pathogenesis because it's still my first love. So you always go back to your first love, which is microbes. I have also shifted my program to look at substance use disorder because of where I am, the role of microbiome in health and disease, and then immunity at the maternal-fetal interface. And I'll kind of explain how I ended up in this really diverse, sometimes feels like an octopus, but really fulfilling program that keeps me on my toes. So where did I start? So I finally get this position in 2008, and I really wanted to do something that brought together that love for virology and microbes and my very growing at that point interest in aging and immune dysregulation that occurred with aging. And there were too many people studying flu. Everybody knows influenza virus impacts the very young and the elderly. It's a very crowded field. I did not want to study flu. So I picked a virus that everybody hates, varicella zoster virus, the chickenpox virus, which you may think, like, why would you want to study that? That's not that exciting. It's chickenpox, we all got it, or some of us didn't get it because you're all too young and vaccinated. But VZV, whether you get vaccinated or not, is a herpes virus that will establish latency in the sensory ganglia. So even if you had a vaccine and never had actual clinical chickenpox, you have VZV in your sensory ganglia. And it will reactivate. 30% of people will experience a reactivation event in their lifetime after the age of 65 primarily. And herpes zoster is a debilitating, a really hard to treat disease. So at the time I started my career in 2008, there was no vaccines, and the only therapeutic was acyclovir, just your generic anti-herpes virus antiviral. So what were the blocks? Well, it turns out that there are no really good animal models to study VZV. The way that people were studying VZV when I started this career was a humanized skid mouse model where they were implanting tissues from kidneys and lungs and skin grafts and all sorts of stuff to basically create some sort of humanized mouse where you could get virus to replicate. But what does that tell us about tropism or dissemination or host microbe interactions? You're really limiting the virus to the human piece of tissue that you stuck in the mouse. It doesn't tell you anything about how this virus moves around. And it's a really interesting virus because it's a respiratory virus that ends up going to sensory ganglia. How does it get there? What's carrying it? Does it go on its own? Is it retrograde transfer? Do you need to have a rash first before the virus gets to the ganglia? Can it get there before the rash appears? What are all the mechanisms? There were lots of questions. You could not answer them in this humanized skid mouse model. So I decided, well, I have all this background in non-human primate immunology now, so why not try to figure out if we could have a non-human primate model of varicella zoster virus. There is a virus called simian varicella virus, but it's been studied in African green monkeys at the time that I started this, and African green monkeys were super susceptible to this virus. It caused actually hemorrhagic disease. Well, African greens are also just susceptible to all herpes viruses, so clearly not the right animal model. I did some homework. I dug up some records from 1967 to 1980 on outbreaks of viral diseases in non-human primate colonies. And I found that rhesus macaques were the only species of macaques that never really got that sick. There was no mortality in rhesus macaques. And I decided I'm going to go after that. I asked for virus from this colleague. I did some research, found who's studying this, and I told them what I wanted to do. And they said, they didn't say this, but I would imagine if they were in Kentucky, they would have been like, bless your heart, it's never gonna work. And I was like, well, you know, I'm 25 and I don't really know like what fear, I don't have no fear of failure right now, so can I just still get some virus? And they said yes. And it turns out that rhesus macaques actually was a great model for this. It recapitulated the human disease. You can see there's a rhesus macaque at 10 days post-inoculation. We did an intrabronchial inoculation to mimic the respiratory route. It has a nice rash on its flank. It looks just like the baby next to it. And so we had an animal model. The animals cleared, and when we euthanized them and took out the sensory ganglia, we could find latent virus in them. We had gone all the check marks of the hallmarks of disease. A respiratory inoculation, rash, immune response in the lungs that moved on to the ganglia, latency. And over the years, we worked on what controls this virus infection. What are the elements that control the acute viral infection? So we could do things in this model that, of course, we can't do in humans, but it mirrored some of the clinical data. We depleted the macaques of CD4 T cells, CD8 T cells, B cells, and asked the question, if you lose one of these components, do you get more severe disease? And the answer that we found was really stunning. So it turns out that if you lose CD4 T cells, the monkeys had more severe disease. And that fit with the fact that kids who had A-gamma globulinemia, which means you can't make antibodies, actually had completely unremarkable varicella. So antibodies were really not important during acute infection. But kids that had any CD4 T cell deficiencies were susceptible. And And so that was great for us, again, in the sense that this is a good model to study this human virus. We then started digging into the questions that I asked you, right? Do you have to have a rash before the virus goes out to the ganglia? So the model at the time was the virus gets in, it has to replicate in the skin, and then it hijacks the nerve endings from the skin back to the sensory ganglia. It never made sense to me. Just never made sense to me. There's lots of people who have subclinical infection, and yet they have reactivation. The vaccine doesn't cause disease, yet there was cases of the vaccine strain reactivating. So if the rash was needed to have the seeding of the sensory ganglia, then how are those people with no disease getting reactivation? And so these are things you can do in an animal model that you can't do in humans. So we actually were able to euthanize animals at different time points post-infection. And it turns out by day 3 post-infection, which is a time where the animals had 0 rash, this is day 3 post-infection, there was tons of virus already in the ganglia. So the virus was finding a way to get into the ganglia within a couple of days of infection. It did not need the rash. How is it getting there? What, like, how is it getting there? And then the other super surprising thing to me was there were tons of T cells in the ganglia at day 3. And for those immunology aficionados out here, maybe there are none, but T cells need, because they're the Navy SEALs, they don't get activated right away. A good T cell response needs at least 7 days to get off the ground. It just needs a really long time to get off the ground. And you want that because you don't want renegade T cells going around killing tissues. So what are those T cells doing there at day 3 post-infection? Could they possibly be carrying virus into the ganglia? Because our lungs are full of T cells. Our lungs, not that of a mouse that doesn't see an antigen because they're specific pathogen free. But our lungs immediately are full of alveolar macrophages and T cells. And so we were able to show by histology that there were T cells dotting these neurons at day 3 post-infection. And there's lots of evidence from other herpes viruses where T cells interact with neurons, and in that synapse, they are able to pass viruses through a synaptic transfer process. So we're able to show that, yes, it is the T cells. The T cells are the Trojan horse in this model. They have lots of virus. They can sustain viral replication because when we did transcriptomics at the single open reading frame, so single gene of every gene that virus encodes, which is 68 plus 4 duplicates, we can find transcripts associated with every one of those genes in the T cells. We also showed that there was this really interesting process happening in the lungs at acute post-infection. Even though there was no clinical manifestation whatsoever, they were getting engorged with a lot of these T cells, and some of them had these homing receptors that would send them out to ganglia. So we were able to show that. We then took it over and used more recently single-cell transcriptomics to show that there is a very significant remodeling of the transcriptome of the infected T cells relative to non-infected T cells, so bystander T cells, that would facilitate the ability of those T cells to get to the ganglia. And so we were able to put this, I'm putting pictures of my trainees sprinkled throughout. I'll call them out at the end. But so we were able to put together this really great model to understand VZV pathogenesis. It's a respiratory pathogen. It hijacks T cells. T cells get into the ganglia because they're transcriptionally remodeled to allow them to enter tissue. Then when T cell pressure goes down, there could be reactivation, and we could trace that too. So one of the other things that I really wanted to do is, what can we make a vaccine for this, right? So I'm thinking, at the time we had Zostavax. It was a crappy vaccine. Like, the efficacy of Zostavax was just as good as if you just never got vaccinated. And so we thought, well, since T cells are both the Trojan horse but also important for resolution, is there specific T cell responses that we're losing with time? And so we compared, we made overlapping peptides, because I told you T cells recognize peptides. So we made overlapping peptide libraries for every single gene that this virus encodes and measured T cell responses against every single viral gene during acute infection, and then we waited months, and then during latency. And we asked the question of which T cell responses are we losing the most over time? And we identified about 4 viral genes. One of them was ORF68, it's glycoprotein E. So we made a couple of vaccines expressing that. So that's, if you look at the graph that says viral loads. We made it as a plasmid, DNA vaccine, we vaccinated monkeys, we challenged them with the virus, and we can show that if they got a hepatitis B vaccine, which is irrelevant, they had robust viral replication, but if they got any of those 4 proteins made in different pools, we were able to reduce that viral load by 3 logs. Well, around the time that we were putting all of this together, Shingrix came out. And do you know what's in Shingrix? Glycoprotein E with an adjuvant. So even though I did not beat GlaxoSmithKline, that's fine, they've got bigger guns than me, it was really rewarding to see how this model could continue to teach us about this virus and that we were just right there with them. So I did get an experience in writing the SBIR and making a DNA vaccine that, you know, it was all very good. So what did I learn from this chunk of time of my life? Just the importance of translational science. What are you doing? Why are you doing it? How is it? Who is it impacting? The collaborative partnership, guidance from sponsors and mentors. I had Ravi Mahalagam send me my first batch of virus, even though he thought in his head this was just a losing situation. He still sent me the virus, sent me every protocol I needed. Nothing in return, just handed it all over to me. He's like, I'm doing this and these monkeys and they keep dying, and if you've got a better idea, more power to you. I'm trying to pay it forward. I try to help as many people as I possibly can, because if it weren't for him and Don Gilden, I would not be here today. My postdoc mentor tried to take claim and credit for the model that I developed. He actually refused. I put him as a middle author of my first paper, and he threatened to write the editor and tell him that I stole the data from his lab, which when you're a first-year assistant professor, it's really scary. Everything was on my side. I had the notebooks to prove that this was all done in my lab after his departure from the university. But Don picked up the phone, who is chair of neurology at University of Colorado and Denver campus, and basically told my prior postdoc mentor to, go do something. And that he was gonna basically stand by my side. So find those sponsors, find those collaborators. They're everywhere. People wanna do the right thing. They wanna help you do the right thing, build your community. And don't be afraid to do things that are crazy. It's okay. The worst thing that can happen is it fails, but you learn something. So continued on my journey with aging. We did some studies with some other collaborators to show that hormones actually play a really critical role in this. So it turns out that menopause and andropause are a real thing. And losing those hormones actually does reduce the immune system's ability to fight infection or respond to vaccines. And reconstituting these hormones would be beneficial as long as it's done in a very controlled manner. I still don't know what the exact mechanisms by which this works, so it's an open project for somebody. We also wanted to further understand why are older individuals more susceptible to disease. And part of it is, if you look over under the left-hand side of the slide, you can see in those graphs, or maybe you can't, that there are 2 lines, and the bottom one is always the older animals. They're just generating lower T-cell responses all the time. And we know that there's issues with the T-cells as we get older, learned that from my PhD work, other people have shown that too. But what I did not realize is if you look at the heat map just to the right of those graphs, you'll see this block of red genes that are really predominant in the adult and doesn't exist in aged hosts, and then vice versa. What you see that's really bright and that's really high expression in young animals is a lot of type 1 interferon response genes. Those are your first line of defense against any viral infection. Aged cells are not able to generate that type of response. On the other hand, the next block of really red genes is acute phase response genes. A lot of interleukin-6, a lot of TNF-alpha. They're great pro-inflammatory genes, but when you're making that much that early in the infection, it's not great. It changes the barrier function, destroys your epithelium, opens up a whole bunch of fluids to come in, which is why older humans and non-human primates end up with pneumonia after a flu challenge. So it also taught us this balance, like maybe the T-cells are not functioning well, but there are major issues in that first line of defense. We're starting off with the wrong pro-inflammatory response. So how can we change that? How can we change that balance and tip it back to having a better type 1 interferon response? That was true in another RNA virus, chikungunya. This was at the time where there was a massive chikungunya outbreak. I was running around. And this is also true in the context of a bacterial infection. So this is non-tuberculous mycobacteria. It's everywhere. It's an environmental bacterium that's in the soil, it's in the water. You shower every day, you're inhaling NTMs every day, but because you have a nice, healthy immune system, you're okay. Unless you're over the age of 65, and then you start having colonization and disease. And this is mediated by mechanisms that we don't fully understand. A lot of people thought, well, it's because older people have more COPD, or they have more bronchiectasis, so inflammation of the airways. That is why this is happening. And so Hadi, again, went back to the fact that we have a non-human, so the mouse model for this just causes disseminated disease, no pulmonary disease whatsoever. So again, paired up with a colleague, we thought, well, let's ask if non-human primate model could be the answer to this. And it turns out that yes, non-human primates will have disease that looks very much like human, and just like human, older macaques will have more severe disease. And once again, we're showing that this is due to this really dysregulated inflammatory response, where they're going more acute phase response, lots of interleukin-6, lots of TNF-alpha, and less this type 1 interferon, less growth factors to promote healing and resolution after you have all this inflammation, and less T-cell responses, which is shown in that bar graph over there. You can see in the young animals, we have lots of lymphocytes that are coming into the picture, but in the aged animals, you have a lot of myeloid cells. So those are your monocytes and macrophages that are just gonna keep making more TNF-alpha and IL-6. So you're calling the wrong parts of the immune system to the equation, and they're just having this self-fulfilling inflammatory response that leads to really severe disease. And so again, how did we start here? When you talk to a colleague and they have an idea, pair up with them. Don't be afraid to go in on a project that, where you're bringing some expertise and somebody else is bringing some expertise. That's part of the fun of science. You don't have to do it all alone. I partnered with 5 different PIs to make this project come to light. We had a pulmonologist on board, we had an infectious disease specialist on board, we had a bacteriologist on board, because I'm not one, and we had a radiologist on board to read all of our scans independently. Bring the team together. Science is so much fun when you have people to do science with. And start small. You don't have to hit a home run every time that you write a grant. Start small and build upon that. I'm going to go on a tangent for a second, but one of the things I'm not enjoying about baseball anymore is the fact that there's 3 outcomes when people go to the plate. They either strike out, they hit a home run, or they walk. It's super boring. I know, maybe none of you remember this. Chad, do you remember the time where there was strategy in baseball and nobody needed to hit like 100 miles an hour ball? Like there was actually like finesse and strategy and sacrifice and bunts and people got on base. Okay, you can do that in science. You don't have to have a hit run. Be the fun team. You don't need a Nature or Science paper every time. Get together with some friends, do some great science, publish some cool papers, and move the field forward, even if it's in scientific reports. Okay, so back to that whole viruses thing, because I keep coming back to that. Once in a while, I'll go in a different direction, but I always come back to viruses. We know that we live in a world where lots of emerging and re-emerging viruses. Didn't mean to scare anybody, just kind of situational awareness, people. So my husband sent me a picture. He was very proud of himself. He's in the Navy, has deployed to Ghana, sends me a selfie sitting in a bat cave. I'm like, you're getting a Marburg test before you come into the house. That's not cool, dude. So anyway, but that's what happens when you study these viruses is you start losing some fun of your life because you're thinking about all the things that you're exposed to. But you also develop a healthy respect to some of these pathogens. So one of those groups that I have a lot of respect for is the filoviruses. So this is Ebola, Marburg, tiny, 20 kilobase RNA, negative strand. They can't even make positive strand without their own polymerase dependent, like they have an RNA dependent RNA polymerase. So you'd think these things would be extinct, but instead, they would kill 90% of the people they infect within 10 days. And so really fascinating to me. And over the years, in parallel with all the programs I was doing, studying why do these viruses cause such severe disease. And then it kind of, every time I try to have a T-cell angle in my project that ends up with something to do with the innate immune system. So it turns out it's all about monocytes again. It's all about monocytes getting hijacked by viruses and making them make the wrong type of cytokines. So again, Ebola virus, story repeats itself. Shuts down type 1 interferon, turns on IL-6 and TNF, which then recruits more monocytes that can get infected, and then monocytes can travel around the body and infect other organs. So spent many years working with fabulous colleagues. My friend Andrea and I started when we were both postdocs, and now she's a tenured faculty at Rocky Mountain Labs. And Heinz, her mentor, who's been an amazing sponsor for both of us. But we, for 10 years, we've worked on this VSV-based vaccine platform. It takes advantage of the fact that vesicular stomatitis virus is also a negative strand RNA virus. You can take its glycoprotein and pop in the glycoprotein of any filovirus, and it makes amazing vaccines that work with low doses, very low doses. So that's great for any developing country because you can actually distribute a lot of doses. And we showed that this is mediated by an antibody response, and we kind of delved into the mechanism of how this vaccine is so powerful. Heinz introduced me to 2 other big Ebola players, and with them, we were able to dive into why does Ebola interfere with the interferon response. So showed that this is all actually mediated by one protein in this virus, VP35, which actually is an RNA polymerase cofactor. So it's important for recognizing RNA to initiate the genome replication, but also at the same time, it shuts down the ability of the host cell, our cells, to recognize that this is a negative strand RNA virus. It doesn't belong here, we need to shut it down. So really cool work that leads to potentially making a VP35 knockout virus to use as a vaccine as well. So more avenues for new things. So everything is humming along beautifully, and then there was SARS-CoV-2. And when life gives you lemons, you make lemonades out of it. So my lab at UC Irvine at the time was one of the very few labs that stayed open. My students did not want to go home. They wanted to do something. And so we did a lot of stuff. We built a biobank of samples. We grew virus for the hospital to use positive control. We helped them when the CDC primers annealed to each other and did not actually give you a result you were supposed to. We tested masks. We did all sorts of stuff. And it was great. It was this amazing experience for my trainees. None of them left home. There were coyotes on UC Irvine campus. That's how few people were coming to work. But we were there, and it led to a whole bunch of papers. So what I learned from the era of SARS-CoV-2? Scaffold onto existing partnership. Other people were also willing to roll up their arms and get to work. Be proactive. I turned on our BSL-3 that had been dormant for 10 years. I pivoted with my trainees. They all graduated with 5 papers because we just all got together. We did it safely, but what they needed was a purpose and not to be home. And I provided that for them. And I'm very proud of that moment because I had to fight so many administrative rules about this. But since nobody else wanted to work, we just took over an entire massive lab. It was great. I had to cede the space back when I left, and it was kind of like, really? And so, okay, I'm cognizant of time. I'm going to tell you a couple more last-minute stories that also came out of just meeting people. So alcohol use disorder, when I was actually a brand new assistant professor, I got approached by, I'm just going to fast forward really quick, by this wonderful scientist, Kathy Grant, who was a neuroscientist, a behavioral neuroscientist, wanted to understand addiction and mechanisms of addiction, and had a monkey model to understand that. So she's very focused on the brain, circuitry, all the things I did not understand. I just nodded, yep. But I asked Kathy, do we know anything else about, other than addiction, what are the consequences of drinking heavily? And she was like, I don't know. And so we did some literature search, found out that, well, in addition to just, this is one of the biggest drivers of mortality and morbidity in the country. It's also one of the biggest drivers of increased susceptibility to infectious diseases. So people with alcohol use disorder end up in the ICU more often. They can't respond to trauma in the same way. And they experience more osteoporosis. They end up staying in the hospital longer if they're admitted. They have more increased rates of nosocomial infections. So on the back burner, we were starting to look at what does alcohol do to the immune system, and built this on smaller grants that led to big grants, and multiple graduate students and postdocs got into this. And so it's been a fun journey to try and dissect what's happening. And the first paper we published was to confirm, yes, so it's hard in humans to pinpoint that it's alcohol use disorder because there's a lot of other comorbidities, and substance use disorder is multi-substance use disorder. But we were able to show in the monkey model, yes, there is actually a deficiency in their ability to respond to a vaccine. And then again, a whole body of work spearheaded by many amazing people in my lab that showed that, to my chagrin, it was not the T cells. Once again, it's the monocytes that were dysregulated, making those two darn cytokines that I keep talking about. Maybe one day I'll figure out, like, how do we overcome this TNF IL-6 axis of evil. And then I had trainees in the lab who became interested in other things that I did not feel comfortable with. They wanted to study microglia, osteoblast, bone. And one of the big things you learn when you get out there is that you got to let your trainees sometimes pick their own adventure, and you can sit on the sidelines. And so my trainees picked their own adventures, and they were able to demonstrate some really amazing things, that alcohol does rewire microglia into a more activated but yet tolerant phase, that it does change the balance between osteoclasts and osteoblasts in a way that leads to increased bone fracture and reduced bone density and strength. And so just embrace the opportunities for growth. Don't stay stale. Encourage and support your trainees when they go off. They want to go off the reservation a little bit. That's okay. And commit to that lifelong learning, because it's really critical. And so I always tell, like, what is the biggest lesson you've ever learned over the 20 years that you've been leading your lab and leading department? And it's just hire smart people and just get out of their way after you resource them. And then the last story of that resonates with that is immunity at the maternal-fetal interface. There's this big theory called the developmental origin of health and disease, where we know exposure in utero can really change the trajectory of somebody's life and susceptibility to disease later in life. We're, of course, an immunology lab, so we're going to be very focused on immunology. And to my surprise, I did not know this until I started reading the papers. The placenta is a massive immune organ. There's tons of T cells, macrophages, NK cells in the placenta that mediate tolerance, fetal growth, homeostasis, angiogenesis, all the things that you need for this growing, amazing interface. And so we started by looking at obesity and the maternal-fetal interface, precipitated by really this colleague of mine. We were both in a junior faculty, mid-career faculty leadership course, and she was telling me about, she's an OBGYN, she was telling me about all the complications she was seeing in her patients who had a BMI greater than 30 when they started their pregnancy. And we got together, put our brains together, and had trainees together who tackled what was happening in the placenta, what was happening in the immune system of the offsprings. And again, started off a trajectory that then set us up to look at other things. Like, again, when SARS hit, we just pivoted. We're going to look at SARS infection during pregnancy. So we know that is a massive comorbidity that impacts pregnant women. So how does it do that? What are the challenges that could be encountered? What about placental transfer of antibodies? Could this be something that we need to look at to encourage vaccination? And then we moved from that onto opiate use disorder, because we need to align ourselves with what the Commonwealth needs us to do, and that is the opiate crisis. So again, being able to show that there is dysregulation of the immune landscape in the placenta with a lot of these exposures that lead to heightened inflammation, poor angiogenesis, reduced fetal tolerance, and a lot of complications when it comes to timing of labor. And then we went back to that. Once we developed a way to develop microglia, we went back to it and asked the question of, we know that there's neurodevelopmental outcomes that are unfavorable in the offspring of women who are exposed to opiates during pregnancy. Why is that happening? So now we're tying our findings in the bench to findings that we see out in the clinic, and trying to pinpoint, are there biomarkers of neurodevelopment that we can identify early on so we can intervene. And so what are the lessons we learned from here? Define and communicate your core purpose. What is your why? Your why can't be, I have to get another paper. What is your why for doing what you're doing? That is really critical. Prioritize research that is informed by your community and what your community needs, whatever that community may be. Understand how basic science can actually inform health policy, and be an advocate for science. Don't be afraid to take a detour and grow your village. I'll come back to this. I think you've noticed the theme in my slides. I've only written one R01 as sole PI, and that's because my first chair made me do it. He told me I would never get tenure if I didn't have an R01 with just me on it. And I was like, fine, I'll write one. I got it. Never again. It's just way more interesting to do science with other people. So follow your passion. Surround yourself with people who will guide you, who will tell you when you've gone off, who will sponsor you, who will mentor you, who will coach you, because those are all very different things. Pay it forward and mentor others, your postdocs. There's undergrads around you, graduate students, staff. Pay it forward. Somebody got you to where you are. Pay it forward. Lead by example. Conduct yourself in a rigorous manner, integrity, ask tough questions, don't be easily discouraged, and give that example to all the trainees that are coming behind you. Challenge yourselves. Try new things all the time. Get out and about. Tell people this all the time. Stop hanging out with your lab people. Look at this. My whole lab is together at one table. It's like a flock of freshmen in college. They just all move together. Get out. Go meet other people, other labs. Make new friends. See what they're doing in their labs. Could be a really interesting way for you to take your science to a new level. Take care of yourself and your loved ones as well. And have fun. It is the best time of your life as a scientist. Takes a village. I love pictures of my lab. I think it's hilarious. We love elevator pictures, so there's a bunch of those. There's my lab over the years. This is Isaac, who was trying to flip Cool Whip into his mouth, and he succeeded. And I think that was the final test he had to do before getting into med school here. But Isaac was an undergrad in my lab at UC Irvine, followed me to Kentucky, where he was a lab technician for 2 years, and is now an M2 here at UK. Brianna also came with me, and she now manages my lab. I wanted to highlight my Ilhem group down in the bottom. These are women I went to leadership class with. We always call each other, so grow your village. They're for different institutions. So I can say, what do you do at Tulane? Because here at UK, this is not working for me. We're perfect, of course. And then funding. I can't do it without all the funding that I've been blessed to have over the years. And so look at that. I left 10 minutes for questions. All right. Thank you all for your attention. I'm happy to entertain any questions about science, about life.
I've got a question. So, first, well, I've got a comment and a question. First, I can tell you that all of this is true, and I can attest to this. So, Ilhem and I went to Jurassic Quest together. Okay? So, when she was talking about the alcohol research, Kathy Grant is a big name in the field. Like, I went to the Research Society on Alcoholism for many, many years doing my alcohol research, and so that's a name that came out to me. But what I can tell you is my wife is an alcohol researcher, and Ilhem invested in her relationship with my wife and my family, and we met up at Jurassic Quest, and they actually still collaborate and work together, right? So all this is real, and so I think it speaks to investing in your village and growing your village and talking to people, even tangential, collaborations. I think it's important, and following through and being meaningful and significant. And so that's my comment. And so my question is, how do you do that, right? So like, how do you decide to be out there and, what's your driver for that piece, for really kind of like paying attention to all the relationships that you build and being like, okay, I've got an idea, I've got an idea. Do you have a secret for that, or is it?
Oh, tough question. Well, okay, so it helps that I'm just an extrovert. I embarrass my 8-year-old all the time. He's always like, my God, do you have to talk to everyone? So that does help. But I think the reason why I love doing that is I just find science fascinating, and I find everyone's work to be really interesting. Actually, the less that it's like my work, the more interested I am in learning about what people are doing. And it's a genuine interest in science because it is something I truly love. Which is why I say, do what you really love. Find your passion. What are you really passionate about? It's not to say that every person you interact with is going to turn out to be an amazing collaborator, There are studs and there are duds in life. And so, you know, sometimes you just have to experience the duds to know which ones are the studs. There's lots of people that you encounter and then you're like, that was great, I'm never gonna work with you again. And there's people you have a 20-year relationship with. And when you find those people who resonate with you, who share your core values, you work well together, just nurture that, be genuine about it, reach out to them. They can read your papers, they can hear you go through your thought, they can read your grant, they could be that person that is outside your field that you get input from. It's just important to be genuine and to just invest in them, just like any relationship you have in life. It takes work. Yeah. Quiet crowd.
Thank you so much. It was a great talk. I have a question about this thing that when you said like lead by example and lead with the integrity. I have seen like when we lead by example and we do the right things, sometimes people around us do take this thing in a wrong sense, and it is considered that you are showing arrogance. It's not like integrity, and it is considered wrongly associated with arrogance. Have you ever seen this thing in your life, and how did you deal with this?
Yes. So when you're pointing out things, right? Like if you say, well, I don't really think you had all the controls you needed for that one experiment. So my advice is always, there's multiple ways to say the exact same thing. And so maybe saying, where are your controls? May not get you where you want to be. Or I'd love to hear more about your experiment. So could you walk me through what you did here? And then you can say, well, why did you did this part? Like, I'd love to learn. And, oh, do you think you would have, like, do you think having this would have helped? Or, so there's ways to ask there's just, over the years, I'm also have the habit of being like a bull in a china shop myself. So I've just learned that sometimes you can't come at things this way. You got to come at things this way and that way. And it takes a little bit of practice. And so it's good that's the other reason you have friends, is you can rehearse on each other. I do a lot of, if I said this, do you think, can you gut check me? Am I off? Like, is this, my husband is probably tired of, I make him read all my emails, almost any email that could be like contentious. I have my husband read it. He's a pulmonologist. But no, just somebody outside the situation. Like, this is what happened. This is how I'm responding. Does that seem like, and a lot of times he'll say, well, it seems like you have some other chip on your shoulder. You're bringing that into this conversation. When I read just this first email, this does not warrant this. So what else is going on in your head? And then you're like, well, it's the third time they did da. So, well, you should have addressed it the first 2 times, but this is not the place, So talk to, that's the other reason for growing that village, right? Having that ability to say, how can I say this in a way that's not that doesn't come across as attacking, but comes across as being collegial and helpful?
Thank you.
Of course.
Hello, hi. Thank you very much for sharing your experiences so openly with us. I see one thing that is common is that you say pivot whenever necessary. So there are 2 things, right? One, if you keep doing something for years and years, something is going to open up for you and people say, okay, that person didn't leave his goal or what he wanted to do forever, he or she, and that's why they are so successful in their career. But some people say, okay, there is this opportunity that you have and you got to grab that opportunity and that leads you to places somewhere else. In your case, you seem to have pivoted and have, grabbed those opportunities. But how does one decide? Do you feel that in your heart? I know it's an emotional factor as well, but how do you know that you want to continue to do something or you want to pivot? Like, what should be the criteria if you have the trick or tip to share with us?
Really great question. And some of it is going to be instinct and some of it is going to be the reality on the ground. Right, so also I'm going to bring up my husband again. He loves to tell me that the definition of madness is repeating the same thing over and over again and expecting a different result. Yes, okay, but we're scientists, right? We like to be like, well, maybe if we tweak this one thing, or maybe if we tweak this thing. So I would say, don't pivot for the sake of pivoting. I already had, for example, I'll just bring the SARS, just COVID era, right? I already had this deep interest in emerging viral diseases. I'd worked on Ebola, I worked on monkeypox, I worked on chikungunya. So for me, pivoting into a new emerging virus was not something that I had to really force. It was not a forced pivot. It was something that scaffolded onto an interest that I already had. The circumstance was here. I wanted to step up. I wanted to do something. I had funding. I did not want it to go dormant. I did not want to be home. I had a 6-month-old and a 3-year-old. I did not want to be home. I love my kids, but like there's limits. I had available daycare because my husband is a pulmonologist and critical care physician in the Navy. So so I chose to take that love I had of emerging viral diseases, marry it onto my projects that were already happening. I was already doing pregnancy and obesity. There were women showing up at the ICU with COVID. It was like, I'm good at this. I ran a BSL-3 before. I know how to handle this. And I'm already doing a pregnancy project. So I'm going to pivot into this opportunity. I was at UC Riverside, and UC Irvine invited me to apply for a job. Right? I've only been there for 4 years. I did all the things that nobody was supposed to do. I followed my PI around. He tried to steal my project. I stayed in the same institution. And I only say that half jokingly to say that there's no set path to how you succeed in life. So you have to follow opportunities and your passion and try to align those two things together. So they called me and they were, would you be interested in applying for this position? We're opening this. We want somebody with experience in high containment to do it. And UC Riverside was building a BSL-3 for me at the time. And it's like, do I leave? Like, I came to this place and they've already started building this thing. And do I leave? And what is the cost of leaving and uprooting my lab? But I sat there and I did the pluses and minuses and the pros and cons and decided to make the move. It came with a promotion. It came with a BSL-3 that was already filled. I was pregnant with my first kid. I did not want to deal with a brand new facility. So there were a lot of calculations in that. So I'd say don't pivot for the sake of pivoting. Don't pivot into something you hate. But don't shut the doors. Like if your first love is to have a 10-year track career at an R1 university and Lafayette College comes calling with a position. So it's an R3, but you can write small NSF grants, you can write R16s, you can do summer research, more teaching. Do you hold out for the R1 institution 10-year track position to come up? Or do you take a chance on this? And those are going to be very personal decisions, but don't close the door just because that may not be what you set out to do. It would be my only advice.
Thank you so much.
Well, yeah, thank you.
Thank you.